encapsulations, we sandwich this pillar plate into a deep well plate for static organoid culture, or we sandwich this pillar plate into a perfusion plate for dynamic organoid culture. So once you created the organoid, then we can utilize these organoids for in situ organoid analysis. We use this for real-time imaging, functional assays, disease modeling, or for the compound screening. So I'd like to briefly introduce the bioprinting techniques that we are using currently. This is the 3D bioprinter that we have in the laboratory. So we load the pillar plate in the machines, and then load the cells in the disposable tips. And we have a robotic arms that goes and pick up these biological samples. We can essentially print 4 different samples simultaneously. And then machines can basically dispense these cells in hydrogels on the pillar. We can print the single droplets, or we can print multiple droplets on the same place. After cell printings, the plate can be inserted in the fully automated microscope, and we can analyze these tissues in situ. And the cell culture can be done in the CO2 incubator in a dynamic conditions. And then using these pillar perfusion plates, we were able to generate multiple mini human organoid, which include liver tissues, liver organoid, liver tumor organoid, using biopsy sample from patient. We also created brain organoid, intestinal organoid, and colorectal cancer organoid. So using these technologies, basically we can create any organoid in the body on these platforms. So if you compare our technologies with the other competitors, these pillar perfusion plates are the only products that allow miniature human cell printing and dynamic organoid culture in high throughput. We can load the cells extremely fast on the pillar plate by using microarray 3D bioprinting or manual stamping. We can also reduce the cell death and enhance the tissue maturity by rapid diffusion in the perfusion wells. Tissue can be analyzed, tested with the compounds, stained, and imaged automatically. And then we could generate the large number of the organoid on the pillar with the small cell culture media volumes. And the operations are very user-friendly because we don't need to use pumps and tubes for dynamic organoid culture. And the pillar perfusion plates are compatible with existing equipments, the cell fluorescent microscope, and also the microtidal well plate readers. And this is a 3 different method we typically use for loading the cells on the pillar. So as I mentioned earlier, we can print the cells suspended in hydrogels on the pillar. And also we can use stamping methodologies. We have side wells and slits on the pillar, and that allow us to load the uniform volumes of the hydrogen droplet on the pillars. And then these pillars can be sandwiched onto the ultra low attachment well plate, and then have this spheroid transfer directly on the pillars. That is the spheroid transfer methodology. And after loading the cells on the pillar, these pillar can be sandwiched onto petri dish with the lid for static and dynamic organoid culture. It can be sandwiched into a deep well plate for static organoid culture, or it can be sandwiched into a perfusion plate for dynamic organoid culture. We have published recently 2 papers, one in the Advanced Healthcare Materials and the other one in biofabrication. So if you are interested in learning more about these technologies, you can look up these papers. And this is the overall process of cell loading and analysis. So this is basically the stamping methodology to load the cells from the pillar. We can have a cell suspensions in this loading plate, and then have these pillar sandwiches stamped on the loading plate, and then after gelations in the petri dish, you can have these pillar inserted in the petri dish with the cell culture media for long-term organoid culture. These are some of the unique features of our technologies as compared to other commercially available platforms. So these pillar perfusion plate allow us to do organoid culture. We can utilize the biomimetic hydrogels for organoid culture on the pillar plate. It allows dynamic cell culture, multi-tissue connections, convenient media changes. It's compatible with the 384 well plate formats and allow in situ cell imagings. It utilizes very small assay volumes for cell culture and analysis, and it's high throughput compatibles. And this is a schematics of the pillar plate and picture of injection molded pillar well plate. So we first created 384 pillar plate and also complementary 384 deep well plate. So pillar, after loading the cells, pillar can be sandwiched into this deep well plate for static organoid culture. And we have generated 36 pillar plate with the 6x6 arrays of pillars. That 36 pillar plate can be sandwiched into 36 perfusion plate with the 6x6 arrays of perfusion wells and 2x6 arrays of reservoirs where you can add the cell culture media. So after sandwiching, we can load these perfusion plates on the digital lockers and then have a bi-directional flow generated under these pillars. This is the last products that we generated. So as you can see, 144 perfusion plate contains the 12 by 12 arrays of pillars. And also complementary 144 perfusion plate contains the 12 by 12 arrays of perfusion wells and 2 by 12 arrays of reservoirs. So we can add the cell culture media in the reservoirs, have a bidirectional flow generated. One of the other unique feature of this platform is that you can actually have multiple different organoids connected by fluidically by using this perfusion plate. And we have done some flow simulations in the perfusion plate. And as you can see, by tilting these plates, you can generate forward and backward flow. And the flow rate is close to about 10 microliter per second, as compared to other conventional microfluidic devices. The flow rate generated in our devices at least about 100-fold faster. And then the second thing I want to emphasize is that we can generate rapid flow within the perfusion wells because of the pillars sandwiched into the perfusion wells. According to the Bernoulli principles, when the fluid passes through this narrow cross-sectional area in a pipe, flow rate is accelerated. So because of these phenomena, we could maintain very fast flow rate in the perfusion well, which is very unique as compared to other perfusion systems. So we hypothesize that we may be able to reduce the cell death in the core of the spheroid or organoid due to mixing under these pillars. So with that hypothesis, we first tested it and see if we can reduce the necrotic core. That is very common problem in the organoid culture and the spheroid culture too. So we created 3 different conditions. The first one we created spheroid in the ultra low attachment 384 well plate. And then second conditions, we created a static culture of the spheroid in the pillar perfusion plates without the rocking. So it's a static conditions. Third conditions, we generated the spheroid in the pillar perfusion plate and start the dynamic culture in the digital lockers. And as you can see, we track the changes of the size over time. And one thing you probably notice is that in condition 1 and 3, the spheroid grow really fast during the time, and it grow up to about 1 millimeter in diameters. And second thing we notice is that after the cell culture, we measure the cell viability of on different conditions in different conditions, and surprisingly, we have seen much lesser cell viability in condition 1, which is spheroid culture in the ultra low attachment plate. So with that data, I was wondering why this is happening, and asked my students to perform live and dead cell staining. And we basically stain these cells with the calcium AM, which is a dye for the live cells, and then with the ethidium homodimer, which is a dye for dead cells. And as you can see, the spheroid culture in the ultra low attachment plate, the condition 1, you do see a lot of dead cells that died in the center, which is a very common problem in the field, which is also called the necrotic core. But on the other hand, if you grow these cells in the dynamic conditions in the pillar perfusion plate, we could almost completely remove these necrotic cores. And we have measured the fluorescent intensity of the green dyes and the red dyes. And as you can see, in condition 3, you could see much higher green colors and much lower red color. Again, indicating that the usefulness, the use of these dynamic culture. And with that idea, second, the question we had was, what if we can create these human organoid in the pillar perfusion plate? Can we improve the cell viability and also maturity of this organ? So we follow the most advanced liver organoid differentiation processes, which is published by Dr. Takebe. And it takes about 25 days to create the human liver organoid from the induced pluripotent stem cells. At day 7, when we create the Fogo cells, we load these Fogo cells either in the 24 well plate in a matrix domes, or we printed these Fogo cells on the pillar plate. And then we follow the same differentiations and maturation processes. So first we check and see if we can generate the Fogo cells successfully. We were able to generate 96% of FOX8 2 positive cell populations, which is an indicator of the Fogo cells. And then this is the entire process of the experiment. So after we generating the Fogo cells, we detach the Fogo cells and then suspend it in matrigel, which is biomimetic extracellular matrices. And then print these Fogo cells in matrigels using 3D bioprinters on the pillar plate. And after gelations, we sandwich this pillar plate into a deep well plate, and then continue the 18 days of cell differentiation and maturations in the 384 deep well plate. And after that, we perform the organoid analysis, real-time imaging, functional assays, disease modeling, and compound screening. And first question we have was that whether or not these bioprinted Fogo cells can be healthy and viable, because you probably heard about the shear stress and the pressures that can damage the cell viability. So We, after printing these cells on the pillar plate, we measured the viability of the bioprinted Boger cells using, again, the casein AM and additive homodimers. And as you can see, all the cells printed show the green color that indicate that there is no dead cells because of the bioprinting. And we measured the viabilities of manually dispensed cells and also bioprinted cells. And as you can see, there is no differences between these, the bioprinted cells and the manually dispensed cells in terms of cell viabilities. And then the bioprinting was very accurate. So we obtained the CV values, the coefficient of variations within 18%. Typically, when we obtain the CV values below 25%, those assays or the cells can be useful for the high throughput screening. And then we created the human organoid in the 24 well plate with 50 microliter matrigel domes. And then on the pillar plate, we created the same human liver organoid in 4 microliter matrigel spots. And as you can see how the morphology changes over time. After 25 days of culture, you can see the circular human liver organoid with the lumens in the center. Same thing can be obtained from the pillar plate. You can see small dots with the black hollows in the middle. And after creating these human liver organoid in 2 different systems, we compared the maturity of these human organoid generated. And one striking thing was that the human liver organoid created on the pillar plate shows much higher maturities. And that was confirmed also by using immunofluorescence stainings. You can see that we have stained these human liver organoid using hepatic biomarkers, HNF4 alpha and bimentin are the most famous hepatic biomarkers. ALB represent albumin. So you can see that we could generate multicellular liver organoid using these differentiation processes on the pillar plate. And these liver organoids secrete albumins. That is the most representative proteins secreted by the liver. And then we were able to generate the liver organoid on the entire pillar plate. This is the stitched images of the 36 pillar plate. We can see that all the pillar contains the liver organoid. And we measured the cell viability from the liver organoid. And then again, the CV value was between 11 to 16%. And we also measured the albumin secretion from multiple different trials. We have tried these 3 times and were able to obtain similar levels of albumin secretions. And also we measured the expressions of cytochrome P450-3A4, which is one of the most representative drug metabolizing enzymes. So we were able to obtain the CV values of about 22%. And liver typically dumped bile acid into the bile ducts. So we wanted to check and see if we can have these bile acid transportation functions in the liver organoid. So we use the green fluorescent bile acid and see if this green fluorescent bile acid can be accumulated in the lumens. And as you can see, the green color continuously increases over time, which is an indication that bile acid transportation function is active in the liver organoid. And with that, we test and see if we can obtain the ICP values or the dose response curves using the model compounds. We have tested sorafenib and tamoxifen. and see if we can obtain the similar ICPP values from the multiple trials. We have done these assays 3 times using liver organoid created on the pillar, and we're able to obtain the similar ICPP values. So this basically indicating that the liver organoid can be used for drug screening as well. Next, we also test the hepatotoxicity of the acetaminophen. This is a very famous painkiller used in the US and worldwide. So we measured the IC50 values of these hepatocyte, the acetaminophen using human liver organoid. And the IC50 values we obtained was about 4 millimolar, which is actually quite close to the IC50 values we typically obtain from other assays using primary hepatocytes. And we also test and see if we can measure the regenerative capacity of the liver. So typically, when the livers are exposed to toxic chemicals, after stop treating the patients with the drugs, the liver is supposed to be regenerated, so we can have these liver tissues regrow over time. And we wanted to test and see if we can demonstrate that regenerative capacity of the liver using human liver organoid. So after treating these liver organoid for 3 days with acetaminophen, we let this liver organoid recover in the expansion media and see if you can observe the regrowth of this liver organoid. And you can see that in the bottom panels, the liver organoid, in fact, regrow after 10 days of recovery in the expansion media. And another thing we notice is that we can see very high expressions of these inflammatory biomarkers overexpressed. So these IL-4, IL-6, and TNFA, these are all the inflammatory biomarkers. So we see that these after the liver exposed to these acetaminophen, the inflammations in the liver was increased dramatically, and during the recovery phase, those expressions of inflammatory biomarkers reduced dramatically too. So we can utilize these liver organoids for these kind of drug compound testings. Finally, we expose these liver organoids to fatty acid and to simulate the steatohepatitis. So you can see that the body P, which is a specific dye for staining the fatty acids, and we can see after oleic acid treatment, the liver organoid became green fluorescent, which means that there is accumulations of fat in the liver organoid, and that was also observed using the fluorescent microscope. So you can see that in the controls we did not expose the liver organoid to fatty acid, but for the liver organoid exposed to the oleic acid, you can see huge enhancement of the green color, which is indications of fat accumulations in the liver. So with that, let me conclude my presentations. So we demonstrated highly functional human liver organoid in the perfusion plate. This could be potentially used for predictive screening of compounds, and we could do disease modeling with these human organoid created on the pillar plate. I'd like to also mention that this is cost effective because we can only use 10 to 100-fold smaller volume of cell culture media. And it is highly flexible and user-friendly. because cell can be loaded manually or using 3D bioprinters, and we can perform static and dynamic cell culture without using pumps and tubes. And we can easily do multi-organoid connections using these perfusion plates. We can do in situ real-time high-content organoid imagings to study the mechanistic toxicity of these drug candidates. And finally, our platform, high-throughput screening compatibles, is built on footprint of standard 384 well plate. Thus, it is compatible with existing high-throughput screening equipment, such as microtiter well plate readers or the confocal microscope. That's it for my presentations. If you have any questions, I can take that questions.
Okay,
Okay,
doctor.
doctor.
First
First
of
of
all,
all,
thank you
very
very
much
much
for
for
your
your
intervention.
intervention. I
I
found
found
this
this
technique
technique very
very
interesting
interesting
and
and
the
the
attention
attention
in
in
this
this
area.
area. Especially, I
Especially, I
can
can
see
see
an
an incredible, very incredible horizon on possibilities that this technique affords to this area. So now let's make a space for a question that public, and if as you want, as you know, you can put them in the chat or can turn on your microphone to ask to this expert.
Okay, so one of the questions I see in the chat box is that what is the what's inspiring to start this merging technology into one? In fact, we've been working in this 3D bioprinting for almost 2 decades. I started this 3D bioprinting back in 2001 when I was postdoc at Rensselaer Polytechnic Institute. We've been trying to commercialize these 3D bioprinting techniques a lot, and particularly in the field of drug discovery. Initially, we print the cells in hydrogels on the glass slide, and then also we moved to micro pillar chips and trying to develop these 3D culture cells for drug screening. But after long uh, 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 developmental processes, we realized that the cells grown in spheroid is not sufficient to really mimic the tissues in the body, and also we could not solve the problems of the necrotic cores over time, because typically to recapitulate the tissue structures in the body, it takes a long time to grow the cells and differentiate those cells. And I show you one example of liver organoid. It took us almost 1 month to create the liver organoid. Therefore, during the culture, cells in the core can be dead, and we needed to prevent those cell death. So that was one of the main reasons why we needed to incorporate these fluidic systems in high throughputs. And then spheroid again is not sufficient. So eventually we have to use the stem cells to recreate the tissues in the body. And therefore we needed to combine these 3D bioprinting to rapidly load and accurately load the cells in the device, and also grow these stem cells into tissues, and also create the dynamic cell culture conditions to mimic the circulatory systems in the body. So eventually I realized that while I'm doing this research, I realized that we need to actually combine these three technology into a single systems to really use this beautiful technology for drug discovery. Yes. So the second question was the result of the drug experiment. We are still in quite early stage, but we were able to recapitulate the data from the primary hepatocyte. That is the cells isolated from patient. And also, I show you briefly about the regenerative capacity of the liver, which was demonstrated using human liver organoid. That was actually typically done in animal models. It's very challenging to actually create the regenerative organoid. Liver is obviously the regenerative organs. So we were able to generate the passageable liver organoid and also demonstrate the regenerative capacity of the liver. Most of the people currently using liver organoid that cannot be regenerative. So this has not been actually published, but we are working on and trying to publish this data in a journal. So that we are ultimately trying to replace the animals using these lab-grown tissues in a high throughput device. So another question is, how do you see the future of these techniques in daily use of bioprinting? So again, the ultimate goal is my research goal is to speed up the drug discovery processes. So for example, I envision that, let's say if you can generate the human organoid in the microfluidic devices, in a high throughput microfluidic devices, you can use these lab-grown tissues for drug discovery. For example, we can actually get the cells from patients, and then, for example, you can recreate the tumor tissues in the patient body using these high throughput microfluidic devices combined with the 3D bioprinting, and then use that tissues for personalized medicines. So you can screen the drug combinations or the drug candidates against these lab-grown tissues, and then find the optimum drug combination for particular patients. And we can utilize those drugs for patient treatment. Okay, any other questions? We actually, I didn't mention anything about the other techniques, but we are currently trying to combine the machine learning and the AI to better predict the patient outcomes. So essentially, whatever experimental methodology you use in the laboratory, those are all in vitro data. But we have to treat the patients in the hospital, right? So that there are in vivo data. So basically, what we need to do is predict the in vivo data using in vitro data. So there is a huge gap between these two. So we try to basically link these two different data using machine learning and AI. So we do analysis of the RNA sequencing data from the patient and try to find the link between this patient data and the organoid data.
That's pretty interesting.
interesting.
Again,
Again,
thanks for
thanks
your
for your
participation.
participation.
That
That
will
will
be
be a
great
great
speak.
speak.
And
And
now I have
now
no
I
words.
have no
I
words. I
just
just
gratitude for
gratitude for
your speak.
your speak.
Oh, no problem. Thank you for giving me this opportunity.
Okay,
Thanks to you.
thanks to you.
And
And
now
now
I'm
I'm
gonna share
gonna
to
share
everyone
to everyone
the
the promotion video of Pan
Pan-American.
American.
Somos Editorial Médica Panamericana, la editorial líder en edición y distribución de contenidos especializados en ciencias de la salud de negocio, publicaciones, formación y soluciones digitales. Contamos con más de 2.500 títulos publicados. Nos avalan más de 70.000 horas de formación a distancia. Presencia internacional. Estamos en los 22 países hispanohablantes y en Brasil. Buscamos aliados estratégicos que impulsen la educación integral de la salud. Al iniciar un convenio, Editorial Médica Panamericana descuentos especiales a los miembros de la asociación. Editorial Médica Panamericana asume todos los costos de la asociación, la publicación de libros, la generación de cursos y programas educativos y otras actividades de formación continua y actualización profesional bajo la modalidad online o impresa. Editorial Médica Panamericana acompañará a la asociación en los eventos que realice, otorgando excelentes beneficios a sus asociados durante el evento. Editorial Médica Panamericana hará difusión en sus redes sociales a sus más de 40.000 contactos del convenio establecido y de los eventos que la asociación organice. La cobertura será a nivel nacional. Los afiliados podrán adquirir nuestros productos a través de la página web con envío gratuito y en nuestras oficinas. Contarán con varios medios de pago y fácil financiación para los productos de formación continua. Juntos llegamos más lejos. Editorial Médica Panamericana. La salud, nuestro proyecto editorial.
¿Vas a empezar a preparar el examen MIR? Te presentamos ProMIR, un nuevo entorno virtual de aprendizaje que te ayudará a obtener los mejores resultados donde y cuando quieras, sin necesidad de desplazamientos. Con ProMIR, el estudio se realiza de forma simultánea al entrenamiento de las habilidades necesarias para superar con éxito el examen. Seguirás un itinerario formativo personalizado, con una metodología estructurada en 4 fases que te ayudará a optimizar tu esfuerzo. Este nuevo y revolucionario sistema de preparación para el MIR cuenta con contenidos enriquecidos y personalizados. Podrás subrayar contenidos, incluir anotaciones y marcadores, e imprimir los materiales y tus apuntes siempre que los necesites. La plataforma contiene más de 30.000 preguntas, que incluyen las de los 12 últimos años de la prueba. Al mismo tiempo que estudias, podrás poner a prueba tus conocimientos con entrenamientos tipo test y simulacros de examen. En tu camino hacia el MIR, contarás con la tutoría y asesoramiento de profesionales expertos en formación médica, que resolverán tus dudas y te orientarán para mejorar tu técnica de examen. Además, contarás con videoclases de apoyo al estudio. La metodología de ProMIR se adapta a ti en función de la convocatoria a la que te quieras presentar y al momento en el que ingreses en la plataforma. Se estructura en 4 fases: calentamiento, construcción, consolidación y competición. Dependiendo de la fase en la que te encuentres, ProMIR te indicará cuáles son tus objetivos de cada día y adaptará los eventos a tus necesidades y a tu ritmo de aprendizaje. El sistema inteligente analizará tu evolución, tu nivel en cada asignatura, el tiempo que has dedicado al estudio y al entrenamiento e irá adaptando los contenidos para maximizar tu esfuerzo y mejorar tus resultados, a modo de entrenador personal. ProMIR es un entorno virtual de aprendizaje totalmente vinculado y multisoporte. Así podrás estudiar donde y cuando quieras, desde cualquier dispositivo. Ponte a prueba. Consigue tu acceso gratuito y descubre por fin una nueva forma de preparar el MIR. Infórmate en www.medicapanamericana.com/promir. Promir, un proyecto original de Editorial Médica Panamericana.
Bueno,
Bueno,
con esto podemos
podemos dar
entonces por concluida
a
nuestra
nuestra primera jornada de evento. Ahora debo hablar en español.
Muchas
Muchas
gracias a
gracias a
todos
todos
los
los
que
que
hicieron
hicieron
parte
parte
de
de
esto
esto
y
y
espero que
espero que pues les hayan quedado varios conocimientos de nuestros increíbles ponentes. Los esperaremos mañana pues con la segunda jornada de charlas. Me disculpo por todas las fallas de logística que hubo durante el día de hoy, pero vamos a mejorar para el día de mañana. De nuevo, muchas gracias
y
y
nos
nos
vemos
vemos
después.
después.
Adiós.
Adiós.
Gracias.
Sí, sí.